They're a real pest because they're the tiny little flies that hang around your fruit bowl. And what this question is asking is it's saying that you've got two different genes that we're going to track in this one. You've got a gene for body color and a gene for wing shape. You can either have gray body or you can have ebony. Ebony is black, right? So they come in gray or black, the fruit flies. And their wings can either be long or vestigial. Vestigial just means basically short. They're like really short. So the two pairs of alleles are located on different chromosomes. We'll see how that has a relevance shortly. But what it's asking is determine all the possible genes of the progeny's offspring of the following cross. Gray body, long wings, heterozygous for both. So we're going to do our 8-step method here now. So let me just scroll this up. If you want to just try and keep one step ahead of me, then that would make a lot of sense. So well, I'll get us started by saying, and it's really important with these this 8-step method. With any cross you do, I strongly recommend that you use the 8-step method. But it's not really that useful unless you've labeled each of the steps. That's really what they want. You've got to treat these crosses as labeled diagrams when it comes to the exam, okay? So just to practice, just force yourself to do the 8-step method so that it's like second nature, okay? So the first thing you're going to write down is you're going to write the parent phenotypes. Okay, and the parent phenotypes in this case is the gray body. I'll just put gray, long. Okay, and we're crossing that with an ebony vestigial. Vestige for short. Okay, so this is the setup. We know it said all we can figure out even, but because they've told us that it's heterozygous for both, one of the parents, this first parent is heterozygous for both. That means it must be big G, small g, big L, small l. So that's the genotype, parent genotype. Okay. Just going to open my chat, and do please, if there's anything that you want to just run over quickly again, or you didn't get the first time, or you have a question about, just shout, and I'll see it in the chat box. I have it open here beside me. Okay. All right. So now we can hopefully guess what the second one is. If it's ebony, we were told at the beginning that big G is dominant to small g for ebony. So the only way it can be ebony, your knowledge of genetics tells you, is that if it's two small g's. And similarly with wing shape, small l is vestigial, and the only way it could end up actually having vestigial wings is if it's two small l's. Okay. So this one, I'm just going to change that small l. That's not particularly good there. Just going to change that to be a. Okay. So you've got to be real careful with your lettering here as well. So the next step is, once we've got this set up, I mean, the rest should be kind of plain sailing. This is often the most difficult part, is getting this step 1 and 2 right. They give you a story. They include something about phenotypes. You've got to deduce the genotype to some extent, and then you're on your bike. So the next stage is meiosis. And meiosis is basically just arrows to show that the different combinations of gametes that you can get. So gametes is what you're actually listing for step 4. So let's list them. Gametes are traditionally put in circles. I really advise you to put them in circles. They don't, some examiners are very kind of old-fashioned and fussy and just, you don't want to annoy them, right? So put the gametes in circles, strong recommendation. Mendel's second law says that it is equally likely for big G to combine with big L as it is for big G with small l. So big G with big L, just as likely as big G and small l, which is just as likely as small g, big L, which is just as likely as small g, small l. And similarly, Mendel's second law applies to all these situations, but in this case, there is only big G and there is only big L. So there's only one possible outcome of gametes from this second parent, it's small g, small l. If you can think of another combination, let me know, but I think that's it. So these are the full available gametes in this fertilization event, which is what we do next. So label it as it is. It's fertilization. Okay, so you do the fertilization. And in our world, for genetic crosses, fertilization is the Punnett square, right? So and in this case, there's only one gamete on the left-hand side. That's the small GL. That's this one here, right? So this one here is this gamete here. Okay. Okay, so you need 4 slots here to figure out what the products of fertilization are going to be, right? The possible zygotes, right? So the gametes that are available from the other parent are big G, big L, big G, small L, small g, big L, and small g, small L. Okay, so when you then put these through the mill, the fertilization mill, you get big G, big L, small L, big G, small L, small L, small g, big L, small L, and small everything in for the last one. So there are 4 different possible genotypes. Now, you see them in the Punnett square, but the next step is going to be to list them. And what are they? They're the progeny or the offspring genotypes. And I would really recommend that you do these notifications of RT news out of the way. What are the genotypes? Well, they're right there, right? So, but I would still, like I said, I would still list them. You've got big G, small g. I'll just list them really quickly. Because that way you can fully label them. Otherwise, it becomes just awkward to label them. You're trying to point to things and it can be not clear. It needs to be 100% clear. This is 100% clear. Step 6, progeny phenotypes, and you list them. There's no way that the examiner can claim that you weren't being clear. What do you think is next? Well, what the question asked, let's just remind ourselves what the question asked. The question asked, determine all the possible genotypes and phenotypes of the progeny. Now you're thinking, hang on a second. So we actually didn't have to go through all that stages 1 to 5. If we just knew what I could have done a back of the envelope thing, or I could have done in my head, and I could have written down all the genotypes right there, because I knew how this is going to pan out. I've done enough of these, you say to yourself. Definitely in June anyway, you'll have hopefully done enough of them that you can see all the possible crosses and the outcomes, right? Because you've done them so often. You've got to still show it all. And I'll tell you why, because marking schemes, and they can vary from year to year, but regardless of how the marking scheme is set up, there's always big marks for a cross, okay? So where the marks are going to be, there's no way of knowing. Unless you've seen the marking scheme, it's impossible. And even if you start off, even if you did see the marking scheme, by the time they finish correcting, they can have changed the marking scheme. And they do that every year. The marking scheme starts out as one thing, and then they decide it's going to be something else by the time the end of the summer comes and they've all finished correcting, all these examiners. So you really have to watch out to make sure that you cover every possible thing. And that's why this 8-step method always gives you everything that you need. Once you've done this 8-step method, all the answers are in there. Okay, so you may just all you may need to do is go and just label, like, part 4 right there, answer right there, part 5, answer right there, that kind of thing. So in this case, the question was specifically give the possible genotypes and phenotypes of the progeny. So we've given the progeny genotypes, and then you write down the phenotypes. And remember, it's going to be gray, long. Which one was the one that was dominant again? Gray, long, yeah. So if it's going to be gray long because it's heterozygous for both. This one is gray, but it's vestigial. This one it's 2 small g's, so it's going to be ebony, but it's going to be long, and it's going to be ebony, and it's going to be vestigial. So you see that they're all possible for phenotypes. I just should have put my label in there, progeny, phenotypes. Now, the last step, I said it was the 8-step method. The 8 is usually a ratio 1 is to 1 is to 1 is to 1. There's one of everything. Okay, then that's just a recap then of making sure we're able to do our 8-step method. So I want to talk about, I'll just shove this down here for a minute. I want to talk about chromosome diagrams. So just have a look there. Just going to new share. So this is a situation a bit like we just did, right? It's very similar, but so it's a round yellow crossed with wrinkled green. We're back to peas again, away from fruit fly. And what we've done here is rather than just like write letters, big R, and big Y, small y, we've put in chromosome diagrams. So what I'm going to do is I'm just going to flick back to I'm going to flick back to our ebony and ebony fruit flies. I'm just going to show you real quick what the chromosome diagrams for this would look like. So the chromosome diagrams, if you were doing this, you would have that one parent would be big G, small g. So these are homologous chromosomes, right? I can do better than that. I'm trying to make it small enough so that you guys can see, but you've got the cross in front of you. So actually I'm going to zoom in here. And yeah, okay. So the first one, first parent, these don't have to be, they can be ovals, is absolutely fine. So these are homologous pairs. And it said in the question, remember, that the genes were located on a different pair of chromosomes. So the gene for shape was on a different pair of chromosomes. However, on the same chromosome, at the same position, you have different alleles, or you can. And in this case, you do. It's a big G on one, but it's a small g on the other. They are both the same gene, but they're just different versions of it. Similarly, over here, you would have a big G, you would have a big L, and at the same position on the other chromosome, you have a small L. So that's one parent. The other parent is this situation where the alleles happen to be the same. Okay? So another pair of alleles with L's on them, and they're the two parents. And this is the cross. Okay? Now, when you're doing the gametes, and you're doing chromosome diagrams, so the chromosome diagram for this one will be, remember, each gamete receives one of a pair of alleles. So from the first pair of homologous chromosomes, you get a big G from one of them, and we got a big L from the other one. Right? From the second one, I'm drawing very badly in this. You can do better with pen and paper. We had big G and small L. So during gamete formation, each gamete receives one of a pair of alleles. So each one is getting a G. But also during gamete formation, either one of a pair of alleles, so say small g in this case, is equally likely to combine with either one of another pair of alleles, say big L in this case. But that's just as likely as as small g with small L. So there's your 4 different types of gametes there from that, but drawn as chromosome diagrams.